Oropharyngeal cancer is on the rise and is a growing public health concern. The most common cause of oropharyngeal cancer is human papillomavirus, or HPV. Viral genes E6 and E7 code for proteins that disrupt cell growth, but alone do not cause cancer. When HPV infects our cells, it can insert its own genetic material into our DNA. But it's not that simple. HPV doesn't just add genetic material. It destabilizes the genome, rearranging pieces of DNA like mismatching puzzle pieces. Scientists sequenced the DNA and RNA inside oropharyngeal cancers and found that genetic chaos triggered by HPV insertion recombined bits of DNA into fusion genes, including FGFR3-TACC3 fusions, or F3T3. Gene fusions are created when two or more genes are abnormally joined together. By studying what fusion genes do inside our bodies, we can better understand why cancers develop and how to treat them. The scientists hypothesized that the F3T3 fusion would be sufficient to cause cancer in cells that also expressed viral genes E6 and E7. When laboratory mice were given only F3T3 genes or E6-E7 genes, they didn't develop cancer. But when mice were given F3T3 and E6-E7 genes together, they soon grew cancerous tumors. Normally, E6 and E7 proteins are unstable and quickly break down inside the cell. But when expressed together with F3T3, E6 and E7 proteins were more stable and lasted longer in the cell. Alone, these genes are unlikely to cause cancer. Together, they make a dangerous alliance. This research shows that HPV insertion can trigger changes in the structure of cellular DNA that can promote cancer development. With this new information, we can start deploying new forms of treatment to counteract the creation and function of fusion genes and help in our fight against cancer.